All right, good afternoon, everyone. My name is John Hudak. I'm the Director of the Office of Cannabis Policy and your moderator for today's event. On behalf of the Office of Cannabis Policy, we thank you for being here for the latest installment of OCP's Cannabis Conversations. We're excited to be continuing this series with a cannabis conversation on testing lab standards. Part of the reason for today's event comes from our hearing a lot of curiosity about how the state's testing facilities are licensed, regulated, and monitored. Our goal today is to have an honest conversation about these topics and to help set the record straight. We currently have 4 testing facilities active in Maine's adult use cannabis program. These testing facilities go through a thorough certification and accreditation process with Maine CDC and go through OCP's licensing process as well. Product testing in general, but especially cannabis testing, is a very technical and nuanced topic. We're very lucky to have the team we have in place here in Maine to work on the certification, compliance, and quality assurance necessary for ensuring the integrity of our cannabis testing program. I have 2 great panelists here with me today to help break down the roles of OCP and Maine CDC within Maine's cannabis testing program. They'll help us better understand some of the practices and procedures in place to ensure quality assurance. Christine Blais is the program manager of the Maine Laboratory Accreditation Program at the Maine CDC. This program provides accreditation for laboratories that perform drinking water and wastewater analysis to demonstrate compliance with federal regulations. The program also oversees the certification of cannabis testing facilities in Maine. Christine has been with the Maine Laboratory Accreditation Program for 11 years. Prior to joining the program, Christine worked at the State Health and Environmental Testing Laboratory for 15 years as an organic chemist. While there, she was responsible for the analysis of soil, non-potable water, tissue, and drinking water for contaminants such as petroleum products, PCBs and pesticides, and regulated organic drinking water contaminants. Christine holds a BA in Environmental Science and Biology from the University of Maine at Farmington. Diana McKenzie is OCP's chemist. In this capacity, she serves as a technical consultant for cannabis testing and works to compile and manage OCP's testing database. Prior to joining OCP, Diana worked at the Maine CDC for 3 years as the Cannabis Testing Facility Certification Officer. She previously held roles at the Maine Health and Environmental Testing Laboratory, the Maine Milk Quality Laboratory, and Maine DEP. Diana holds a BS in chemistry from the University of Maine and a master's in soil science from the University of New Hampshire. We're going to start off today with a moderated discussion, and then we'll use any remaining time we have at the end to address questions from the audience. For audience members, please feel free to use the QA function at the bottom of the screen to submit questions throughout our event. Christine, I'd like to start off with a question for you. Can you explain the Maine CDC's role in the cannabis lab certification process?
Well, thank you, John. Thank you for inviting me here today to speak. So first of all, there are 2 people in our program, myself and Felicia Pomiak, who's the other certification officer. So the CDC's objective is to protect the health of cannabis users in Maine. And we do that by certifying the testing facilities, which I'll refer to as labs out of habit, probably for the remainder of this. We certify them so that we can ensure that the data that they're generating is reliable, it's traceable, it's legally defensible, and that it proves that products are safe for sale. The certification process is very extensive. It involves a lot of document review, as well as on-site assessments and analyst interviews and review of all the materials and equipment that go into the generation of data. And so we're enforcing the rules of the certification program through the CDC. And on top of all of that, the laboratories are also required to have ISO accreditation for all of the methods and the matrices that they're required to test before our program will give them certification.
Thanks, Christine. And as a follow-up, what is ISO accreditation and how would you explain it to a non-scientist?
So ISO stands for the International Organization for Standardization, and it's a non-governmental body of global experts that come together and they figure out the best way to do something. And then they come and they create a standard basically that needs to be followed in order to generate quality results. And so they have standards for quality systems, standards for manufacturing, food safety, and also laboratory testing. And that's just to name a few of them. And so any organization that wants to have ISO accreditation would need to apply for that and then meet their requirements, be assessed, and then they would get that ISO stamp of approval. So the ISO standard for laboratory testing is for any type of laboratory testing. And our rule actually incorporates a lot of the standards that ISO would require as well. And just a quick example of what a standard might be would be the laboratories have to check their incubators twice a day, 4 hours apart, to make sure that they're running within the proper temperatures for the method. And the thermometer that they use to check those incubators has to be verified on a accepted standard schedule. So let's say annually they have to verify those, and then those have to be checked against another NIST traceable thermometer. And all of those records need to be kept and made available to us for review. So the ISO assessment requires, again, that application, and they're visited every other year by ISO. And on the in-between years, they have a paper audit. And so having both of those gives like a dual layer of oversight.
And just to reiterate a point that you made, Christine, ISO is not something just specific to cannabis. It's not something just specific to Maine. You've worked in a lot of different labs throughout your career in different parts of state government. ISO is the standard used in labs, period, correct?
It is one of them, yes. It is an accreditation body. It's a standard that laboratories can certainly follow, and it is internationally recognized as a standard, yes.
Thank you for that, Christine. Diana, I'm going to turn over to you next. Can you tell us a bit about your role at OCP and the kind of work you do here to ensure our cannabis testing facilities are operating in compliance with statute and rule?
Sure. Yeah. One of the major roles that I have in my job is to compile and analyze all the testing data for the adult use cannabis program. So all of the testing results from the program are submitted to the OCP from all the labs each week. And I take that data, I review it, and I analyze it to monitor for discrepancies that might indicate a problem with either the program as a whole, with a particular licensee, or with a particular testing lab. For the testing labs, I first look for kind of general errors that might be in the data, such as analysis dates that don't make sense. I check to make sure the fails are properly identified. I investigate analyte results that seem unlikely. For instance, if it looks like there might have been a cross-contamination issue or something to that effect, I would flag that and work with the lab and with the CDC to kind of resolve the issue. On a larger scale, I also look at things like overall potency in flower or fail rates for specific analytes like yeast to mold, and then I run statistical tests on that data. The statistical tests that can show things like biases. For instance, if a lab is always reporting higher potency results for flower than the other labs, or if fail rates for certain tests are always much lower than other labs, those things become apparent in the aggregate data, and we can follow up on them and try to find the source of the problem and eliminate that source. So, like potency testing results are analyzed twice a year by using histograms and trend analysis. Histograms are graphical representations of the distribution of potency results from each testing facility. Once the histograms are constructed for each lab, also by matrix, by flower versus concentrate, they can be analyzed for measures that would indicate if there's a bias present. Things like kurtosis and skewness are two items. So far, through the years that we've been analyzing this data, there have been no indication of bias in the potency results in the adult use program. I also use trend analysis for the potency data, which is simply just graphing the potency results over time by each testing lab, and then looking for sudden or significant increases or decreases So trends for individual licensees are also analyzed so that this can be kind of taken into account when analyzing the testing facility trends. And again, there have been no significant concerns for testing facilities that have been found for the trends in the potency data either. So, and then moving on to something like microbial analytes, such as yeast and mold. So testing results for flour are analyzed again twice a year by using histograms and also by using fail rates. So similarly to potency analysis, the histograms are constructed for each testing facility and analyzed statistically for measures that would indicate a bias in results. And since microbial results are by nature not normally distributed, the histogram analysis for microbials centers around a comparison of the numbers from how many results are reported at just below the fail threshold versus how many are reported at just above the fail threshold. So this analysis will show if the lab is tending to report lots and lots of results at just below the fail as compared to overall fails. So this would indicate a bias, whether intentional or not, to give passes to samples that are actually should be failed. And this method of data analysis did bring to light an area of concern for testing facilities on 2 separate occasions. And OCP, in conjunction with the CDC, we were able to work together and have that source of bias corrected each time. And the other area is the fail rate analysis for microbial contaminants. That's also done twice a year and is a simple comparison of fail rates, either for yeast and mold or for the other microbials. We also do it for pesticides, for metals, but the big one is usually the yeast and mold. We just compare them between all the testing labs for the same time frame, and the results of the fail rate analysis across all the testing facilities for the microbial testing has shown fairly consistent numbers over especially over the most recent 3 years, with any inconsistencies mainly being attributable to the licensees rather than to the testing lab. So from the work that I have done to monitor the adult use testing program, I would say that the labs are performing very well and really do not appear to be introducing any significant bias or faulty results into the adult use program.
Thanks for that, Diana. And a quick follow-up. Part of the work that you do identifies both problems that might exist based on individual test results, something that you might see, something that you might flag, or the overall trends. Could you talk a little bit more about ways in which you identify some of these challenges and how you would flag them internally?
Sure. Yeah. Well, apart from the review that I do of the data for lab performance, I also try to notice when there are trends in certain sample types or from a single licensee. For example, if a licensee is suddenly failing many batches when they were previously doing very well, I might reach out to compliance, and together we might try and reach out and help the licensee track down what the issue might be. Sometimes it turns out there is a piece of equipment that's not being cleaned properly or it's not functioning properly. Sometimes we have found that there are issues with how batches of flour or products are being sampled or how they're being treated before or after sampling. I would say that in each event where I've been assisting with the compliance team to try and locate, figure out what the problem appeared to be, it came down to either the method of sampling being used or the misuse of various forms of treatment being used for the cannabis material, and typically it's been flour thus far. These problems can either happen by, mistakes, happen a lot, or just being unaware of the requirements of the sampling in the proper way. So we want all the samples to be taken in a fully representative way for the overall batch. And unfortunately, the problems can also be due to, purposefully to circumvent the sampling and testing requirements for the products. And that's really something we try hard to find and stop that from happening. So far, in none of the instances where I've been involved with helping out compliance and trying to figure out these problems, where we have found serious sampling or treatment issues that ultimately did result in, cannabis product recalls, there are no instances where the testing lab was at fault. In every event, the testing labs were quite helpful to everyone involved. For instance, by being able to help the licensees with their own investigations into the causes and the sources of contamination at their facility, or by helping walk licensees through proper sampling and testing requirements, and including to helping the OCP with the different ways to test for contamination at a facility.
Great.
So, and just like any system, our cannabis testing program, it relies on all parts of the system, both knowing how to operate cleanly, being willing to make the effort to do so. And I think the vast majority of the adult use licensees do both of those things and do them quite well.
Thanks for that, Diana. That last point in particular, I think, is a really important point to drive home. A reminder for members of our audience to use the Q&A function at the bottom of your screen to send in any questions that you have. We'll get to questions after the moderated portion of today's event. Christine, over to you. How often does Maine CDC assess the cannabis testing facilities, and what are you reviewing or verifying on site to ensure compliance?
So currently we visit the facilities every year, and just for comparison, the EPA requires that laboratories that are testing public drinking water be visited at least every 3 years, and Maine's program is a little stricter and we'll visit them every 2 years. So the cannabis facilities are being visited by us every single year, in addition to having that ISO accreditation as well. Prior to our going out to an assessment, we asked the laboratories to send us all current copies of their method SOPs, their quality assurance manual, and any related quality system documentation for us to review very carefully to make sure that they're consistent with what they had submitted the previous year, to make sure that everything is compliant with the rule. And if anybody's unfamiliar with what a quality assurance manual is, that is the laboratory's guidance document that describes the way the laboratory is going to be managed and how samples are going to be managed. And it includes a large variety of subjects. So our rule actually requires that they have 37 separate components in their quality manual, and those can be found in the rule. And those include things like a policy for sample management, corrective actions, document control, training, things of that nature. And it's very extensive. So we review their quality manual to make sure that they have a really robust quality system in place. And then when we go to the facility, we're looking at a lot of documents and records. We're looking at all of the records of, equipment maintenance and validations and reagent preparation. It's very a lot of records. I think one of the more valuable parts of our visit, though, are the analyst interviews. So we actually talk to the analysts and the laboratory management, ask questions about how things are done. We look at each method that they run. We talk to the analyst and we ask them to sort of walk us through their process, explain what they do, show us where they do it, show us the things that they use. And while they're describing that, we may be looking at the instrument to make sure that they're being checked and that they all have their stickers that show they've been verified. We're looking at the reagents that they're using and making sure that they're not expired. And I think more importantly, we're listening very closely to what the analysts are saying and making sure that what they're describing in their procedure and what they're showing us matches what their SOP says, because it's very important that they document what they do and they do what they say in their document. So we're making sure that those are in alignment. And it also gives them an opportunity to ask us questions. If they're not 100% clear about how to interpret a rule or how to achieve one of the requirements, we're able to explain it to them, what it is that we're looking for. And so it's a great opportunity to sort of have that open dialogue about what we see. And we also are very clear if we see something during the assessment to let them know right on the spot. We don't want anybody to be surprised. So we'll explain why something is a deficiency and give them the opportunity to discuss that with us and have a better understanding of why they're not meeting the rule requirement. While we're there, we're also looking at raw data on the instruments. And so there are times where it can be difficult to have a good understanding of the quality of the data just based on the numbers on a spreadsheet. So sometimes being able to actually look at the raw data on the instrument, see the quality of the chromatography, see the quality of the integrations done by the instrument, and look for things like improper integrations or just signs of trouble is a really valuable thing to be able to do in person on site. That's difficult to do sometimes through just routine data checks. But that is something that we do as a program as well. So each month we'll pick a different required test. So for example, solvents. And we will just arbitrarily pick a date and ask each of the laboratories to submit a complete data package for a test that was done on that day or the next day closest to that. So it's very random. They're not able to pick their best package and send it to us. So that gives us the opportunity to look at the whole package a little bit slower and really have a better understanding of exactly how they're documenting things and how they're doing things in a way that's even better than what we can do on site sometimes. And they'll include all of those records when we ask for them. So at the end of it all, after an assessment, any of the deficiencies that we observe, or if there are things that we say could be a recommendation for improvement or just a simple observation, all of those are communicated to the laboratories, because the goal is continuous improvement. We want the laboratories to do good work, and they want to do good work as well, and everybody wants to be compliant with the rules. So we will communicate the deficiency to them, and then we will offer them guidance on what needs to be done to correct it. And then they are obligated to respond to us with a corrective action and explain what it is they're doing to fix the problem. So it's not as simple as just, thanks, we see we made a mistake, we'll fix it, and that's the end. They have to describe to us what they're going to do to fix it, and then they have to provide evidence of the correction. So if they say they're going to update an SOP, we want to see the update to the SOP and make sure that it meets the requirement before we sign off on it. So it can take a bit of time going back and forth sometimes to resolve any of the deficiencies that we find, but I think that at the end it results in a much better understanding of the expectations and a really solid process going forward. So yeah, so that's in general how we do these assessments.
Thanks for that, Christine. Whenever I'm talking to someone who's unfamiliar with the cannabis industry or is skeptical of it, I always tell people, go into a retail store, go visit a manufacturing facility, go visit a cultivation facility. It's likely going to combat some of the preconceived notions you might have about how the cannabis industry works, that it's actually by and large a state-of-the-art agricultural operation and manufacturing operation. And from what you're describing and from what the three of us have seen, walking into one of these cannabis testing facilities, they're labs like any other lab. They just happen to be testing cannabis, but they could just as easily be testing any other organic matter like we do across the state, across 50 states and around the world. They are state-of-the-art scientific enterprises. And again, I think it's one more part of the cannabis industry that people don't really fully appreciate until they see it with their own eyes, until they visit it, until they experience it. My next question is actually for both of you. What quality assurance and quality control requirements exist to make sure labs are operating compliantly day in and day out? How are these similar or different from the requirements of other types of testing facilities in Maine? I'll start with you, Christine, and then we'll go to you, Diana.
So I'll just quickly describe quality assurance is sort of a process-based, proactive idea to prevent problems from happening. So that includes what I mentioned, the quality assurance manual, a plan for how you're going to prevent things from going wrong, ensuring that you have qualified technical directors and quality assurance officers and analysts that are capable of operating a laboratory like this. Like you say, they're very advanced, and they require people that have the education and the experience needed to properly operate a lab like that. We would want to see records of training that include data integrity training and demonstrations of capability before analysts are allowed to test samples. They would need to do proficiency testing, validation. So all of that is part of quality assurance. Quality control is more related to the process, and it's a way of detecting if something has gone wrong. And so that's what we would call like the batch QC. So every sample that gets tested in a batch would have associated batch QC. So that might be laboratory blanks that would demonstrate that the process isn't contaminated, or lab control samples where a known amount of standard is spiked into a matrix and you have to recover an amount that falls within an acceptance range to show that your process actually works for each batch. Matrix spikes, surrogates, there's a number of different batch QC, and they all have to meet acceptance criteria that are documented in the SOP. And if they don't meet that criteria, then that needs to be investigated and corrected, or if they're not able to correct it or re-extract because they don't have enough sample or whatever it might be, then at the very least that needs to be noted on the certificate of analysis that there was some sort of batch QC failure. The QA and the QC of the cannabis testing program was modeled after the environmental testing program. So the rules that are applied to the drinking water laboratories and the wastewater laboratories. So a lot of those are very much the same as those laboratories would see.
Diana?
Yeah, well, I don't have much to add other than that the quality control requirements that Christine just mentioned are very standardized in various areas of testing, whether it's environmental or food testing, like in milk labs, or biological testing. The use of blanks, negative, positive controls, duplicates, standard reference materials, or proficiency testing. They're just required kind of across the board and in any real accreditation or regulated laboratory testing program, including cannabis.
Great, thanks for that, Diana. I'm going to go back to you, Diana, for the next question. If an issue does arise at a cannabis testing facility, what are some ways OCP and the CDC might collaborate to investigate and/or resolve that issue?
So on my end, typically, if I do note some kind of problem or discrepancy in the data or in a specific test result, I will communicate directly with the CDC, with Christine and with Felicia, to discuss the issue, kind of go over the details, and see what they think about it, what is their opinion on it, and then work together with how we will try to resolve it with the testing lab. So for example, it might be that the lab's not complying with maybe reporting requirements for certificates of analysis or something like that. Or maybe there's something more systemic like a bias in fail rates. In any case, we would discuss how best to approach the lab and what measures both the CDC and the OCP can take to get the issue corrected, and how OCP could assist by doing stuff like, maybe rulemaking is required, or whatever other supportive measures that we can supply.
Christine, can you tell us about the types of records that cannabis testing facilities are required to maintain and why those records are so important?
Everything is recorded. Records are so important for traceability and legal defensibility. So our program adheres to that adage of, if you didn't write it down, it didn't happen. And so essentially everything has to be written down. It's required in our rule that all information that contributes to the generation of data has to be recorded somewhere. So just the list is very extensive, but just to give you a kind of a quick example. So as an analyst, you're about to set up a batch of samples and analyze something, and it might require a bit of a preparation extraction procedure beforehand. And so we want to know, what, when, and how. So we want to know the analyst's initials and the date and time and the method of preparation that they use. We want to know the lots of every reagent and standard that was used in that process that can be traced back to a reagent preparation log if something like that is in place. And it should be if they're preparing things sort of apart from the day-to-day batch QC or the batch prep. They should have a reagent log. We would want to have the equipment ID for every pipette, balance, anything that's used in that process that's traceable back to an equipment maintenance and verification log as well. We would want to know the amounts of everything that they use to make sure that they're adhering to their SOP and they aren't doing anything too creative. We would want to see all of their instrument calibration and batch records. So those would have date time stamps on them usually. And so that allows us to determine that the sample went onto the instrument at a time that makes sense for when the sample was actually prepared, that, you know, a sample's not prepared and then just kind of left in limbo on a bench somewhere for weeks. So we're looking at that. We're looking at the batch QC. Those all have to be recorded. Evidence that everything was reviewed and signed off on by the laboratory management that's approved to do that test or to do that part of it. And then any notations of failures. So if something doesn't go quite right, that needs to be annotated in the records. It has to show that the analyst or the supervisor noticed that something didn't go right and what did they do about that. Even more importantly, there has to be an explanation of any type of record change. And so if somebody writes down the wrong number, you can't just write over the number and put the correct one. You can't white it out. You can't erase it. You have to just draw a single line through it. You have to date it, initial it, and then you have to write an explanation of like why you changed the number. So the original record has to be retained and it has to be readable. So there's nothing that can be hidden in the records. And when a record is required but is not included, so if somebody forgets to put down the date of something or they forget to put down the lot number of something, we're looking for that type of thing, because what happens is you end up with a break in your traceability. Everything has to be traceable back to the original materials used, and so records are just extremely important. It's what helps us to be able to say that we have confidence in the data, because we can see that every step of the process was done properly and it's been recorded.
And once again, to reiterate the point, this is not something unique to cannabis testing, right? This is standards for scientific integrity, that level of record keeping and record making. Otherwise, the scientific process in any lab or even in non-lab settings really can be broken down, and then the replicability is threatened, correct?
Absolutely. Yeah, it's critical. It really is critical. And it's extremely helpful in the case of where later on down the road, something is questioned. Maybe it's 6 months later and someone says, hey, I'm not so sure about that result. Something looks funny about that. Without those extensive records, it would be very difficult to find a reason for why something might be off. We're not always able to say for sure that there's a explanation for it in the records, but at the very least, I can look at the records and say they did everything according to their SOP. I see no signs of QC failure. I see no evidence of contamination. I see no evidence of anything wrong in this process. So it allows us to maybe focus on something else. It doesn't tell us what it is, but it gives us a good idea of what it isn't. And that can be enormously helpful. Without those records, you're guessing, and we don't do that. We don't guess. We have to be sure.
So in a minute, I want to move on to methodology, but one topic that I do want to bring up before we move on to methodology is proficiency testing. Christine, I was hoping, and then I'll turn to you, Diana. I was hoping you could explain what proficiency testing is in the context of our cannabis testing facilities and why it's required.
Sure. So a proficiency test is a blind study where a mock sample is created by an approved PT provider, and that is sent to the laboratories. They're required to do them and pass them before they're awarded certification, but they're also required to do them every year for every test that they are required to do for different matrices. And so the provider will spike that mock sample with a known amount of contaminant, but that is unknown to the laboratory. And the laboratory has to process that PT like they would any other sample. And we will look at the records to make sure that they're doing that they're not, giving the PT its own special spa day and treating it with extra care and running it, many times. Like, we want to make sure they're treating it like everything else gets treated. And they will submit the results of their test to the PT provider. And when the study closes, the provider will send us the results of the study directly, and then they'll also send a copy to the laboratory. So we're looking at the results of the study at the same time. And we can see what the acceptance ranges are and how close the laboratory got to the actual answer. And we're looking at all of that. And in order to pass the PT, they have to fall within the acceptance range. They need to make sure that they're not reporting things that are not in the study. So an example is the pesticide list is very extensive, and the PT providers are not going to send us PT that has every single contaminant in it every single time. And so the lab doesn't know whether something is in it or not. They just know that they have to test for everything. And if they report that something is there that the provider did not add, then that would also be considered a failure, because a false positive is not any better than missing something that is there. So anytime a lab has a PT failure, they need to investigate what the cause was. They need to draft corrective action and share that with our program. And then they need to generate a makeup PT for that failed parameter and pass it. And if they don't pass it a second time, then the lab would be suspended for that particular parameter, that particular target analyte in that test. They would not be permitted to report that until they correct it. And then they have to do 2 passing PTs to get that back. So they're one way of measuring how the laboratories are performing and that their procedures are working properly. It's just one of many ways that we accomplish that, but it is a blind study, so they have no idea what the results are supposed to be. So it can be helpful.
Diana, I just wanted to check in to see if you had anything to add on proficiency testing.
No, I don't. I think Christine is very complete.
So just as a reminder to the audience, use the Q&A chat function, or the Q&A function at the bottom of your screen to send in questions for the question and answer portion of today's event. As I mentioned a minute ago, I'd like to shift into methodology now. Christine, are labs required to use validated methods for each analyte they test? And what's the process for approving the methods used by the labs?
Yes, they absolutely have to use a validated method for their required testing. The methods can come from an organization like the AOAC or FDA or US Pharmacopoeia, but if those methods are not available for cannabis in the matrices that they're trying to get tested, then the laboratories are permitted to develop and validate their own methods. So when applicable, let's say a manufacturer has developed a method for technology and that method has been certified and approved by AOAC, then we would read the reference method, we would look at the AOAC certifications, and we would verify that that technology and that method is able to achieve the reporting limits that need to be met in order to be compliant with the contamination limits that the rule requires. So we want to make sure that the technology is sensitive enough to meet the rule requirements for contamination. We also want to make sure that those methods have been verified in the matrices that the lab plans to use it for. So if they've only been tested for flower, but the lab wants to use it for an edible and it hasn't been validated in an edible, then our program would require that they do their own in-house validations for those matrices. If a lab is developing their own method in-house, then they're responsible for doing the complete validation study. And it's quite a bit of work. They would have to do a detection limit study. So they would have to show us to what level they can actually say that, yes, this contaminant is there. It's not just background noise. So how low can they go in their detection limits in the different matrices that they want to apply for? They would also have to do accuracy and precision studies at different concentrations using multiple replicates to show that they can achieve a certain level of accuracy and precision. They would need to do inclusivity and exclusivity testing for the micro methods, and they would also need to demonstrate that their process is free of contamination. So they would submit an SOP to us, and they would also submit these validation studies, and we would review them and make sure that they met the requirements of the rule, and that it covered all the matrices that they were applying for. And if for some reason we felt that the SOP wasn't adequate, it was missing, critical components of QC, or we felt that their data validation didn't really the detection wasn't low enough, or they didn't use it through the different matrices that they might want, we would ask them to go back and do some more before they would be approved for that method. And so, and the detection limit studies are actually required. They need to verify their detection limits each year because there can be changes in sensitivity of instruments, and maybe a new analyst, or maybe they've modified and updated a procedure. So we would want to make sure that they're able to continuously achieve the detection and the reporting limits that they originally validated the method for. So one thing that's a little bit unique is the validation of potency methods. So because there's so many different matrices that might come in that require potency testing, the labs will generally validate sort of in types of samples. So they'll have validations for flour, concentrate, and edibles. And then within each of those, they may further break it down and have validations for types of edibles, let's say. So a process for extracting and analyzing gummies might look different than a process for extracting and analyzing chocolate. And so they would need to demonstrate that their process works properly. And if a lab were to encounter a matrix that was unique to them, that maybe wasn't working with their validated procedures and they needed to modify it, we would expect that they would verify that it functions and that it's accurate before they start generating compliance data with that. So to answer your question, yes, absolutely. They have to validate and they have to have the data, and it has to be reviewed and approved before they can start using that method for any kind of compliance testing.
Excellent. So my last couple of questions are going to be for you, Diana. A reminder to the audience, drop your questions in the Q&A function at the bottom of the screen. And this last question I have for you, Diana, is actually a question that has come in as we've been talking from some of the audience participants as well. What is normal laboratory variability? And could the same sample see a variance in test results within the same lab or across labs?
Sure. Yeah. So I'm going to start with differences within the same lab. So a lab receives, they have one sample to analyze from a batch of cannabis products, whether it's flour, edibles, whatever it is. They get the one sample. The source of the variance and results for that one sample within that lab might be things like instrument drift, change in calibration, overall small differences in how the sample is prepared between an analyst or between different days. And so with that one sample, the overall variance that is accepted within the same lab in this program is 5%. So they can have a maximum variance of 5% for that sample. So for instance, on Monday, the sample might show a value of 153 ppm for lead. Tuesday, you run it again, and it might be at 149 ppm. That's a completely acceptable variance. It's still showing good accuracy and good precision. So that would not be a problem. So if we move to the differences between labs, then in that case, the largest sources of differences in a batch of product between multiple labs is definitely going to be the sampling. But if we're not talking about the sampling or what happens before it gets to the lab, then, like I just discussed, like the different variances that are going to occur within the same lab will also be happening within, between labs. Every lab will have their own different sets of variances. Even then, so a difference between 2 or 3 or 4 labs Between 10 and 15% is considered acceptable for most types of comparison evaluations, such as a proficiency testing, like Christine just covered in her discussion. So, and reasons for this, that even if two labs are running samples for potency, they may be using different instruments. They may be using different calibration standard producers. They're sourcing them from different companies. There can be many differences between labs that will affect the final testing result. And even if everything is being done exactly correctly per SOP, per all the rules, there's just going to be some natural variation between labs. But the point that I would like to make here is that in our adult use program, so far, the data shows that the variation between the 4 labs for potency analysis, for example, between labs, the variation is less than 5%. So that's comparing all the data in the program and is really an amazing accomplishment for the program as a whole. It indicates that all the labs are performing up to all of our quality assurance requirements, and the data that the licensees are getting back are really as accurate as possible.
And to follow up there, Diana, quickly, you talked about the variability standards that we have within our program, but those also reflect variability standards that exist in laboratory science generally, correct?
Yeah, and I would say that Maine actually was very lucky when we had our adult use cannabis program starting out, because we did have that environmental base to start from, and we just took all of those, really high level quality assurance, quality control requirements, and we just put them right in place. And I think that gave us a huge head start. We're not seeing a lot of the problems that other states are seeing with their testing programs. And I think we can be really thankful that we just happen to already have that in place. And we used what we had in place instead of trying to build it from the ground up.
Well, thank you, Christine and Diana, for all your insights so far. We're now going to take a couple of questions from the audience. For the audience, if you've not done so already, you can submit your question in writing through the Q&A function at the bottom of the screen. And for the audience, you'll pardon me, I'm operating off of 2 screens right now. The first question, and I think this is actually a question for either of you or both of you. As a manufacturer, how do we raise concerns, and who do we notify when we notice inconsistencies that may be an issue, like you've stated during the event? For example, a product or a product type that's passing all the time and then all of a sudden is failing consistently.
So I'll jump in on that one, but so you can certainly bring any concern that you have up to myself, or, I believe the compliance section has a place where you can submit complaints or concerns, and we can definitely discuss it and go from there. A lot of times, if it's really specific to the testing lab, I will either tell that licensee to go directly to Christine, to the CDC, and submit their complaint directly to them, or we can go together and discuss it with Christine and to see what their understanding or take on it might be.
And I'll have the OCP team, I'll check in a second to see if you want to follow up there, Christine, but I'll have our OCP team drop in the webinar chat a link to our complaint form on our website that you can use both for the type of complaint that Diana just mentioned, or any concerns or complaints that you have both in the adult use program or in the medical program. But Christine, I wanted to check in to see if you had any follow-up on that question.
We also encourage the licensees, if they have questions about their results, or if they're unclear about the sample acceptance or sample handling, to also talk to the laboratories, to ask them questions if they're unclear, or if they're not sure about something. give the laboratory an opportunity to try to explain if there's something to be explained. But certainly if you don't feel as though you're getting any traction or not being heard, and that you need somebody to step in and look at it a little bit further, then absolutely, our program is happy to work with the OCP and try to determine what the problem might be. But like I say, we also encourage the people to just talk to their laboratories first and see if there's something that can be determined at that level.
Diana, the next question I have is a question based on an answer that you gave earlier. You touched on licensees purposefully trying to manipulate test results, or I believe you mentioned sampling. What happens when that happens? As a retailer, it's alarming to us that product with illegal pesticides or arsenic could hit our shelves. So we're interested in what happens in these scenarios where it doesn't seem like it could be a simple error, but an intentional deception.
Yeah, well, that is a great question. So it all depends on if they're doing it well enough to stay below the radar so that we don't detect it. I mean, that we can only do so much. We see the data. The rules have a lot of reliance on the licensees doing the sampling themselves, doing it correctly, and doing it honestly. And if that's not happening, there are different ways. So either we don't catch it because we can't, we're not watching everybody all the time, right? Sometimes we do catch it, and sometimes then in that case, if we do either catch it or a pattern is suspicious, then we'll open an investigation. The compliance team will step in and complete a more thorough investigation on what's happening. And sometimes we'll find out what's happening, and sometimes we don't. When we do, then we can issue notices of violation, and then you open up the whole procedure for fines and recalls and other types of consequences for whoever was not doing what they should have been doing.
In any of those situations, Diana, end up resulting in an investigation. Can you talk a little bit about how, with the compliance team, both in terms not only of analyzing data, but also going into the field when investigations are happening?
Sure. Yeah. So generally, if it's something pretty, if we're suspicious that the sampling or the testing is not being done correctly, so generally I will go with the compliance team out to the site to help the procedure of looking around and just have them walk us through what they did. Does it make sense? Does it make sense? Pardon me. From a testing standpoint, does it make sense from a scientific standpoint? What kind of things should we start testing for? We could sometimes we'll definitely take our own samples of the product to make sure it, is it matching what the previous testing showed for this batch or not? Or we will go in and we'll just start taking some tests and just to see where the contamination might be located. So in that kind of way, we can kind of pinpoint what's going on, what has happened. Was it due to just error, or was it done intentionally? And we just kind of pull those threads and see where it leads.
Thanks for that, Diana. The next question, and you touched on this a little bit, Diana, but I think it's worth repeating. This is a question from Randy, who asks, who collects samples, and are sample collectors evaluated or qualified? I often hear lab results are only as good as the sample received.
Yep, well, so that's true. So in our state, the licensees take their own samples. So whether you're a grower or a manufacturer, you are responsible for taking a representative sample of your batch, taking it properly, and submitting it for testing for all of the required tests for that matrix. So I do believe that most of the time, we do have resources out there to walk people through how to take a representative sample, how to make sure your tools are clean, how to make sure you're taking enough product, what kind of sample containers you should use to submit your sample to the labs, and what kind of paperwork needs to be filled out. I mean, there's a lot that goes into it. But we do have a lot of resources available. And I think most people do it correctly and are really trying to do it right. It does turn out that if you do it correctly, you're going to get better results overall for your product anyway. So it's really not worth trying to, again, take shortcuts, cut corners. So I think overall, yeah, that's how our program's set up. So licensees are responsible for their own testing or their own sampling.
And we do have a best practice guide for sample collection. It just got dropped into the chat. But also your compliance inspectors are good resources. If you do have questions about sample collection, Diana has done internal trainings with our compliance inspectors to make sure that sorry, my lights keep going out. So they understand the intricacies and the nuance and to make sure that sample collection is done with integrity. I'm checking the time. We have time for at least one more question. And I think this is a question, maybe we'll start with you, Christine, and then jump to you, Diana. But if I'm wrong about that, feel free, whoever, to jump in. How does OCP ensure scientific comparability when Maine cannabis testing labs report different LODs and LOQs for the same analytes? And are results below a lab's LOD looked at in retests or in regulatory decisions? And if you need me to repeat that, I'm happy to do so.
I can try to answer that. So the first thing that we look at is the contamination limit for a particular test. And so a laboratory needs to demonstrate that they can report below that limit, okay, or at that limit at the most. Ideally, we would like to see them reporting below the limit. There are some target analytes that are especially problematic, that are very, very difficult to analyze. And in some cases, laboratories might have an LOQ or a reporting limit at the contamination limit. That's not very often, though. So we want to see that they're achieving an LOQ below that limit. Then the detection limit is what supports the lab's claim of the LOQ. So when a lab does their detection limit studies, what we would like to see ideally is that they can achieve a detection level that's 3 to 5 times lower than what they're reporting at. So a detection level is simply the laboratory saying that we can see this contaminant at this level, and we're certain that it's greater than the background noise, and it's not a quantifiable value. It's not something that we could like accurately quantify at that level at the detection limit or the LOD. So we would not ask laboratories to report to that level. If a customer says, we want you to tell us if you see it in there at all, then a laboratory could potentially generate like a qualified result saying that we see something that's over the detection limit, but below the reporting limit. So we can't report it, but we can tell you that we see something in there. And it's usually with like a J flag or some sort of a qualifier, just to say that this is not this is an estimated number. It's just giving you information, really. So we want to see those detection limits because that's what sort of holds up the claim for the reporting levels, but we don't ask them to report down to that typically. Does that answer the question?
I think so, and I'm going to check in with you in a second, Diana, to see if you have anything to add to that. But before I do, our team is going to put a poll up here in the Zoom for you to share your feedback about today's event. While we tick down in our last minute or two, we'd appreciate it for you to fill out the survey and offer any feedback that you have about today's event. Diana, any follow-up on that question?
Yeah, I think, and I think, so if you look, at, I'm just going to take pesticides, for example. If you look across more than one lab, and you'll see that different labs will have different reporting limits for those pesticides. And that's based on, to some extent, what Christine was discussing. I mean, there's an MDL study, there's a statistical way to get to what that reporting limit, the MDL, the detection limit, and the reporting limit, which is the limit of quantitation, what those numbers would be. The lab doesn't have to do that. They can, as long as they are meeting the rule requirements and making sure that they are able to report a fail, or can report at least to the half of the reporting limit or the fail threshold, they can do that. So you're not going to see the same reporting limit across all the laboratories. They do have a degree of choice in that matter. So that's not something that's going to be identical.
Excellent. Well, that's all the time that we have today. A huge thank you to our panelists, Christine Blais and Diana McKenzie. Thank you. Thanks all of you for taking your time out of your day, especially on a holiday week, to come and listen to an important conversation about lab testing and laboratory standards in Maine's adult use cannabis program. Please don't hesitate to reach out to us if you have any questions in the future. And as a quick reminder for any adult use licensees on the call, the supplemental budget passed by the legislature included changes to the adult use cannabis excise tax and sales tax rates. Those changes will go into effect on January 1st, 2026, so around the end of next week. And so remember that as you are preparing and paying your taxes. Anyways, thank you so much for attending today, and stay tuned for future cannabis conversations that OCP will be hosting.